Okay, good morning. This is week 12 about manifold and tensor. So this is I would say the more advanced topics in single cell data analysis. Not every typical pipeline will involved in those concepts, but I believe it's important because we are dealing with nonlinear relationship between data points and also high dimensionality. If you have a tensor structure, that means you have a high dimensional matrix. Okay, matrix is 2 by 2, but the tensor will be above. So this is the outline for today. We are going to look at if you have a single data set and how can we use the concept to do this manifold learning. The illustration will be dimensionality reduction. So we already have some of the PCA, t-SNE, UMAP. They all related. But there are some of this embedding algorithm was designed so in between PCA and the t-SNE, UMAP, there are isomap, Laplacian, eigenmap, etc., etc. There are many, many of those dimensionality reduction algorithms, and they are using manifold structure as a concept. Then we will talk about manifold learning alignment. So manifold learning is for apply to a single data set. Then if you have two data sets, for example, from a different modality of the single cell data IC, and you have those different technical approaches to get the data, but from a different perspective, apply to similar population of cells. And you try to align them to learn the relationship between these different measurements and cross the population of cells to cross-validate or discover new patterns. We will discuss a few applications, and then I will mention single-cell attack-seq plus single-cell RNA-seq. This is a very popular approach. If you want to go beyond single-cell RNA-seq, you can do this single-cell attack-seq. It's an affordable approach right now to get the genome level of open chromatin structure information. So we will see how can you combine the information from these two modality measurements and learn something new from there. Then I will mention the tensor and the decomposition. We have applications. For example, we have our own applications for tensor decomposition used for denoise the network information. Okay, so what is manifold? If you search, you probably get some picture like this. If you depend on the domain of the knowledge you search for. But in mathematics, this is an abstract object or a space, which you show this global structure, very complicated. What you the understanding is you have a local Euclidean relationship between data points on those objects. So manifold learning is try to understand those data points relationship, but bear in mind that those relationship between data points are non-linear. But we can approximate the whole structure of those data points as a whole by thinking the neighbors are nearest neighbor between data points follow the Euclidean or linear relationship. Well, PCA and MDS stands for multi-scaling dimensions scaling. they are sensitive to those nonlinear structure. You will see with this complicated global structure, PCA will put the data points superimpose onto each other when you do the dimensionality reduction. The algorithm designed for taking the manifold structure into account will try to avoid those sensitivity, okay? So as I said, the manifold learning problem, this is unsupervised. We don't need to set up any training. And the dimensionality reduction applied to manifold structure is a typical manifold learning problem we try to solve. So after you learn the structure, you reduce the dimension. That will facilitate your downstream analysis, like classification or other task you want to apply to those data points. So let me explain this local or global relationship between data points. So if you have this 3D structure, looks like this. So you know when you construct this structure, you know this data points. In the middle, you have this red, dark red, and to the end, you go through this rainbow color until you have the dark blue. So this is what you know. What you want to reduce the dimension from 3 to 2, you want to fully unfold this structure. So you keep the global structure, you're still in the middle, you have this dark red, and then to the both end you have dark blue. So if you think this is a manifold structure, then you believe the local patch, so all the points with their nearest neighbor, and you can measure the distance between them. And this distance you can trust because they are local distance. They are Euclidean distance. You compute the distance between those neighbor cell data points, and you just rely on those. But you cannot rely on the distance when you jump away from a neighbor. For instance, if the one point from this dark blue to dark red, if you look at the Euclidean distance, this is not too far away, okay? Because you fold the structure, this end close to the middle. But if you follow this local patch movement, you should go from this end to the middle, you have to go through the long way, cross all the spectrum, all the short distance parts by steps instead of jumping. So this is just an illustration of this relationship. We try to use approach which sensitive to try to conserve the global structure to allow you, after the dimensionality reduction, you spread out. And this is the more realistic representation of the relationship between all the data points in there, okay? So when we deal with the single cell data, we have even higher dimension, and we don't know whether the cells in here and similar to cell in here, but in reality, if you want to move the cell from this position to here, they have to transit from a long distance, maybe quite a different development trajectory. Although the final product looks like a very similar cell type, but if you trace back, they may have to go through very long, very different trajectories to make a link, okay? So the idea is to reduce the dimension from a big D to small d, but keep the global structure. That's our manifold learning. Okay, if you search online, there are many manifold learning algorithms, and we only will discuss isomap and Laplacian eigenmaps. They're relatively simple, but there are others follow almost the same idea. So you have this manifold structure on your mind. You have to keep the global structure using different approach. Those ones, using eigenvalue decomposition, so we can easily incorporate it in our class. And hopefully you all know already PCA or others, so we put side by side, you compare what's the difference between them, okay? Again, just repeat myself. So modeling is to find the local neighbor structure first. Try to use these procedures slowly to learn to expand your relationship between long distance data points and from local to global. So here the paper talk about the cellular state space. Okay, so our consideration right now is to consider cells are embedded in the cellular state space, and there are high dimension nonlinear relationship between them. So we use manifold to approximate this structure. So this paper published in year 2000 in Science. So this wasn't a long time ago. I remember year 2000, I was in Sydney and celebrate the new century in the Sydney Harbour Bridge, and people so happy. But that's a really relevant and new approach. But you will realize it's so simple. Just as I described, you cannot rely on the Euclidean distance to do the PCA, to do the dimensionality reduction. You have to consider the so-called, what they call this ISO map. So this should be called geodesic. Okay, instead of using Euclidean distance, you have to use geodesic distance to construct a network first, and using small steps to calculate the distance between data points. Then using eigen decomposition or MDS to expand this structure to learn its low dimensional representation to keep the real relationship between points, okay? So isomap in its core is MDS. The only difference is the pairwise distance between data points. The conventional or standard MDS using straight line Euclidean distance. You just measure Euclidean distance and build a distance matrix. Remember the map between different cities, and you have a travel distance between one city to another. If you have this distance table, you can, on your mind, to map the location of those cities to reflect and to satisfy the pairwise distance relationship between cities. So here is the same idea, but we are not using Euclidean distance to measure the points between the distance between points. We have to use geodesic distance. So how can we get this distance? We will build a network first, okay? Calculate small distance between data points. And then we move from one distance to another. Will be not directly compute Euclidean distance between long distance relationship, but we have to move small steps on the graph, like a worm to cross this complex landscape. You have to move continuously using small steps, and we add up those small steps to replace Euclidean distance. Then the rest of them are the same. So the eigenvectors of a distance matrix will be used as a projection of those reduced dimensions coordinates, okay? So this is a 3D map, and if you calculate Euclidean distance, this is a 3D structure, then you can go through. But in reality, you have to climb onto this hill and mountain valley and move. So this red curve is the trajectory, the path you will measure the geodesic distance. The blue ones will be like three-dimensional. You calculate the distance between 2 points. And in this example, if you have the structure of this horseshoe and you project your points without considering the distance from one to end to another, you get this relationship that tells you A and C are very close to each other, and D and B close to each other, but in reality, AC, DB are equally distributed if you follow the geodesic distance, right? This is an example in the neuroscience. They collect functional MRI data collected from, for example, different individuals or from same individual at a different disease development time, maybe. They create a network or collect this matrix called functional connectivity. That's between different points in the brain, maybe. What you need to do is to measure the similarity or difference between those collected information. So this is a 2D matrix. So you compare between them and see whether this new data set X is more similar to A or B. So this example, if you measure using Spearman similarity, then you look this new participant, a new patient, for example. You look the matrix, look at the structure, the information, the data, then it's more similar to B, because the distance between this X to B, Bob, is smaller. However, if you consider the relationship, the real relationship, consider the manifold structure, replace with a geodesic distance, what they found is this matrix the most similar to A. And they know this because they probably simulated the change using simulated data. Just example, okay, for the application unrelated to the single cell data analysis, but just get the idea. MDS using geodesic distance is isomap. So you start with this distance matrix to replace the values, not using Euclidean distance, but look at the geodesic distance. And once you have this matrix, then the following steps are the same as the standard MDS. So the key part is how can you compute or to get this geodesic matrix, distance matrix. So the most common approach is to build this KN network, K nearest neighbors. So K is a hyperparameter you have to define. If you want to use K equals 4, then each data point will find 4 nearest neighbor make a link. If you say K equals 5, then the algorithm will start by looking at 5 nearest neighbor for each points. Then once you can imagine you connect those and build this KN network, Then you have to apply another algorithm. This is called the shortest distance between network nodes. Okay. So you have cells, and look at nearest neighbor. You build a distance. Could be something like this. Okay. So I just made it up. So you need to find the distance, follow the trajectory. So you have A and B. You need to find the steps. Okay, you have 1, 2, 3, 4 steps. Or you want to go this way, 1, 2, 3, 4. So this is equal steps. This is always 4 here. But you also have to consider the weight. If you have the different weight, you have to consider those values added up. So the algorithm to find the shortest distance on the network from one node to another, this is called the Dijkstra algorithm. So he's a Dutch computer scientist and got a Turing Award because of those contribution to develop an algorithm to find the shortest path between any given pairs of nodes. If you have one node here, another, you need to find the shortest distance. Move from one end to another. Follow this build KNN network. You cannot see, I jump from here to here and jump because there's no direct link between them. You have to move those KNN networks. So once you compute the shortest paths, harmonic steps between all the nodes, okay, you create this pairwise shortest path distance, and then you give to multi-dimensional scaling algorithm. Then you get the results. At this corner this is Julia, another computer language, very similar to MATLAB. So I use this for illustration. They have a package called manifold learning, and you have Swiss row the data set X, and then you just fit and give the name of this method. Then you will get the transformed dimensionality data from X. Okay, from X becomes Y, and Y are low dimensional data, and after the isomap transformation or mapping. So KNN and this graph-based to model the relationship between data points, they are most commonly used, but you can also use so-called data diffusion. We have some examples. The diffusion, you apply got a Gaussian kernel, and this help you to convert the distance. If you have a long distance between data points, this kernel function will help you to, after transformation, it will emphasize the nearest. When you have a short distance, this information will be amplified. When you move away and there's a soft cutoff, then you lost these connections between data points. So I believe some of the algorithm, if you use data fusion, may be more computationally efficient. Because the network, you build a network, and then the shortest distance, the Dijkstra algorithm, may be not as efficient as the data diffusion algorithm. Okay, any questions so far? So we just quickly run through this bigger concept and then first example of manifold learning algorithm. Okay, so this another one is slightly more complicated than ISOMAP. This is published in 2003. So this will also start by build a graph, a KNN graph. Then it uses a slightly different strategy and to translate the problem into a eigen decomposition problem, but apply to this matrix after Laplacian transformation. We will see how this is done. So this is the structure of this algorithm. Okay, so you build a network, measure the Euclidean distance first, and then run through a kernel. Okay, just some kind of soft cutoff, and transform this distance matrix, and emphasize the data which are close to each other. Okay, make those values higher, and for long distance, make those values lower. Then you compute two matrices. One is the D, a diagonal of weight matrix. So basically, this is the degree. Okay, but because the network, the adjacency matrix is weighted, so this becomes a weight matrix for each node. And then remember, how can you compute the Laplacian matrix? That will be the D, okay, degree matrix minus W. So W is original adjacency matrix. So you compute Laplacian matrix once you have the D and the L, and then this translate into a so-called generalized eigenvector or eigen problem. Then you have a L times F. F contains all the eigenvectors, and this equal to lambda, those eigenvalues, and with the D matrix and times eigenvector matrix again. What we need to get out of this eigenvector problem is the F. So F contains eigenvectors. So those eigenvectors are the corresponding embedding you really need for this original data matrix, where you use that to construct a graph. Then through this procedure, and you get the embedding. There's some options when you construct a graph. You look at nearest neighbor using Euclidean distance and to find the edge. So you assign a value to how far away from each edge. And then you keep those nearest neighbor. And in this step, you can select either you want to have a direction or not. So for this edge, indicate the nearest neighbor of this point is this, but not the opposite. And then you can make the symmetric, so you collapse those directions. If there's a link, then you keep that link. Or you can apply, say, I want to use so-called mutual KNN, then you only keep those links which two nodes are each other are the nearest neighbor. Okay, some those links like this and that, because they're not a mutual nearest neighbor, it will be removed. You keep this graph. So there's several different ways to construct this KNN graph. Then you apply a kernel function to transform those distance values. If this distance between I and j are small, then the likelihood or weight to give will be large. So you put one xi in the center of this, and then you look at j. If j is away from this center, then you look at the value of this Gaussian surface. Then you have to be very close in order to reach the higher value. So this function, this transformation, will automatically give you some cutoff. Okay. Change the weight. You have to be very close in order to get the high peak value. If you move away from the center, then the value you after transformation between you will be lower. So this information will be recorded in matrix W. Okay, we can consider this as adjacent matrix to make a network. It's between distance after transformation between I and j. So once you have this W for all the nodes, then you found the degree, okay, the summation of all the weights for each node. Once you have a D, you have a W, you compute the L matrix. That's just a difference between D and the W. Then you just plug in these two matrices. They're build up the built-in function in most of these scientific computing algorithm, these packages, languages. For instance, in MATLAB, you just gave A and B, and then this generalized eigenvalue problem solver will return V and W. And to apply this function, we will give L and D. So A will be L, D will be B. So you give these two matrices, and you will get V and D. V and D are the projection we want to use. A small example, not a very good example though. This is a 2D example. We want to project these 4 data points onto 1D. So put them into a line and see if I put it in line, and this 1, 2, 3 will be here, 1, 2, 3, 4 will be here. Let's look at how this Laplacian eigenmap will solve the problem. So we compute the distance and create a nearest neighbor network like this. So 3 nearest neighbor is 1, so make link for 1, okay? Is that okay? So we have a network. So this network can be get the distance matrix, so 1, 3 connections. Then you have this weight matrix. So this is the network itself. Okay, 1 and 2 are linked, 1, 3 linked, 1, 4 linked, 2, 3 not linked. So this is adjacency matrix for this. And you have a D matrix, you compute how many connectivities for each of the node. And then the difference between them are the. So here, for a small example, we skipped this kernel transformation. So we didn't do anything on this distance matrix as well. Okay, so we get Laplacian matrix, then apply this, you get embedding, okay? And then we project the first node in position 0, and the second node put into -3, put it here position. Third node put it in the coordinates 1, and the 4, and the last node for 2. So that's the embedding after Laplacian eigenmap. You convert a 2D data into 1D, and their relationship are reflected in this way. So this is a very small example. Will be not very intuitive to show the why it is like that, but just show you the steps, okay? So they are somehow linked. You can compare with the PCA. This is for nonlinear embedding. The PCA certainly only applicable to linear embedding situation. And then in the middle, you use, in PCA, we found the largest eigenvectors. And we use covariance matrix. And here, we use Laplacian matrix and use generalized eigenvector problem. So there are some parallel similarity between them, but the information applied here, this Laplacian matrix contains the information of those nonlinear relationships. So why PCA is not a good if you have this data structure and you know you should, after embedding, this red and blue should be separated. And most of this manifold learning algorithm consider this global structure will give you the projection to help you to separate those red with blue, the red at one end, blue at another end, diffusion map. But the PCA will not be able to do that. All right, any questions? So this is a very active field in the data scientists groups. So you can find a lot of online information about the different simulated data in this high complex structure, and then after dimensionality reduction using the correct algorithm, you get something like this. And for this structure, you see, okay, this definitely the PCA only do a good job for those. And this is important for us when we apply dimensionality reduction. If you don't consider those structure, then you either biased your analysis or lead to the wrong direction, or you don't have a good resolution, or the completely wrong answers to get. We have mentioned that in FATE, the FATE using manifold learning in its overall design. So FATE, remember, is in reality is MDS, because the last step of FATE is MDS. So it started by looking at the relationship between data points and build a pairwise distance matrix. So this matrix reflects all the pairwise relationship between all the cells, and then apply a kernel, okay, function to convert that fully connected, pairwise connected network into a KNN-like network, only because after this filtering, you penalize the long distance between data, and basically you chop off the link between those long distance. At the end, you get this KNN-like structure. And then they apply some other so underlying manifold learning calculated. So based on this structure, it start to learn the low dimensional. So once you have this distance, pairwise distance, you can just use MDS to get the embedding. We also mentioned quite a lot of this pseudo-time analysis. They have consideration of the manifold learning in the middle, like this DDR trace and this KNN-based, and those are different algorithms we mentioned last week about the pseudo-time analysis. In the middle, this like diffusion map, they all have a consideration of a manifold structure. Try to learn these continuous steps, move cells from one state to another. So side by side, from the same group of EOS group, and they have another called MAGIC to do the single cell data imputation using same idea as a fate. But in this case, they are not do the dimensionality reduction, but after you build this diffusion or KN graph, they start to learn the neighbor cells and use neighbor cells information to fill up the missing values. What they consider is some of these zeros so sparse our measured single cell RNA-seq expression matrices. But if you look at the neighbor cells and they try to fill up those missing data, they can come up with a more continuous overall structure and fill in all these missing values, give you very smooth overall expression matrix. Okay, so if you gave an expression matrix like this, there's a lot of zeros, give to MAGIC. MAGIC will apply this and learn the manifold structure and learn the relationship between neighbor cells, and they use neighbor cells information to fill in zeros. So at the end, you get a matrix very smooth, almost there's no zeros. People like this because the downstream of this comparison or the DE clustering, a lot of algorithm will be biased a lot if you have a lot of zeros, but will do a very good job when you have the full structure, full matrix. Okay. So you will get like DE results, very beautiful, and the correlation between genes, if you have a lot of zeros, you'll always get 0.2 correlation coefficient, but after the smoothing, these two genes maybe get 0.8 or even higher relationship. So this is a way, if you, in some situation, if you want to smooth the data, it's a good way to go. It's also very fast. And, okay, so let's move to another topic. So we have two matrices. They all generated from data points from high dimensional, and we want to align them. Okay, what do you mean by align them? We want to learn the global structure from one data set, and they have similar global structure with another. We put them together. The samples or cells may not from the same, exactly same cell, but because you know the sample is from same population of cells, it contains the cells in the comparable states. So you want to learn from this technology, you'll get the relationship between cells is similar using another technology. One example is like these two objects. One is a teapot. You rotate it horizontally, and then this toy or person, and you also move in the same way. The move, like sampling from the different position, different angle for the same object, although they are unrelated, but there's somehow this structure between samples, just like this, a group of cells you just measure from different angles. And there's different samples, and you measure from different angles. There somehow internally there's a link, so you can make alignment between this with this. Okay, align the global structure basically. So manifold alignment is a class of machine learning algorithm that produce projections between two different sites or more. You can align multiple sets of data and give the original data set lies on the common manifold. Okay, so you can think about there's abstract project, mathematical, this manifold, but this manifold is just the structure between one to another. This structure is the same. You believe they share the common manifold structure. No single points are related in these two pictures, but their manifold structure are shared. So if you believe that, then you can try to project this to that manifold structure and project this also on that. In that case, after your projection, two data sets together, you found which point, this point will close to this point. This point will be closing to this point. So you align them, and you learn the relationship between these two. Yeah, so after you project data points onto the common manifold, okay, manifold is a latent structure. You don't see them. You don't see it, but you just using this idea to utilize this relationship with each data set to strengthen knowledge between relationship between the two different data points. Yeah, so basically you align them onto the joint latent space. This is a very small example. You have on your left is X, so this is structure. On your right side is a Y, and you align them, project them onto the common manifold structure. It's in the middle. So then you have the one-to-one alignment between them. This is another example. We actually have source code for this. This is like aligned to protein structure, 3D protein structure. You have a data, and that's different amino acids, and then you have the structure illustrated with this red 3D structure form, and then you have another blue. Okay. Then you can imagine you will have a network information, okay, from one to another, which one is linked to another. And then you have this for the blue molecule. Then you apply manifold alignment to that algorithm. This algorithm will put, that's blue and red, Just quickly give you some idea. So you have the red structure in a pairwise distance relationship. And you also have a Y pairwise. So inside this algorithm, we will put these two matrices in this position, diagonal position. And this will create a so-called block. So if you have n by n, and we assume this will also by n, for example, it can be different, okay? Then our block matrix will be the whole thing. So after you put this, so the block matrix will be 2n by 2n. So I'm showing you what's inside the algorithm to do the alignment of these two structures. So it's very easy. You just concatenate two big matrix into an even bigger, okay, 2 by 2, 2n by 2n matrix. And then you introduce an I matrix, identity matrix, where those positions are ones. Yeah, I think so. To make this symmetric, it's ones as well. Yeah, I have to double check, but see whether the ones are here or here. So you just introduce some information to the one-to-one relationship between those points. Then you give this block matrix to the eigendecomposition, and it will give you the projection. So it's pretty simple. So this will be given to eigenvector problem. You will get the projection from two data sets. Then you align. This is the results. So this result is good because you can see them, but for the even higher data points, you don't see them, but after the alignment, you have. So this is one of the early applications. This is another different domain adaptations. This is another manifold alignment applied to data with some different time points collections. But those papers just a little bit early, so the data quality not as good as today we can have. Another approach they call it coupled non-negative factorization. And this idea is to do the non-negative matrix factorization. So factorization means you have a data matrix, but then you decompose them into two small matrices. The multiplication of these two will be the original one. You can do this for separated two data sets, but this algorithm will consider to produce these small matrices, but consider both matrices at the same time. So you cannot freely choose the values you want to approximate to the original data, but you have to consider both data sets at the same time. That's why it's called coupled, and you have this and that at the same time. You try to approximate both. There are some other possibilities, but still, I said those early stage of the, but this field has been developed so fast, and many good algorithms have been developed, also applied to better data sets. So we should move forward to look at a more updated list of the publications. I think we can take a break and then we discuss the multi-omics approach, especially, and today we can have, for the exactly same cell, you have a single cell, a tag-seq, RNA-seq. You don't even have to align them because they are already technically aligned. Okay? All right, let's take 5 minutes break. Okay, so recent years, this multi-omics approach has been become feasible, and you can get other omics information. From in addition to single-cell RNA-seq expression data, you, for example, get epigenomics data for the same population of cell or for the same cell type, exactly same cell. So I want to give you a quick introduction of this epigenomics data, what they look like, where they come from, especially for those without biology background. So I want to make things clear. So the product will help you to, for example, to generate those genome-wide epigenetic information. What we can have right now is more feasible is so-called single-cell ATAC-seq to using 10x genomics solution to get this ATAC-seq, which open chromatin structure information from individual cells. But in order, the computer scientists or people without biology background, you don't know this, I have to go back a little bit. So eukaryote organisms, you have the chromatin structure. It contains euchromatin and heterochromatins. So those are the highly compact, closed structure where euchromatin is open, and you can start to have a transcription or other regulatory functions get involved. And this structure is not constant. It may change for different cell type and different conditions, and some region become heterochromatin, some become euchromatin. So this procedure is called chromatin remodeling. There are a lot of markers getting involved, so you can detect those markers and see, okay, this region contains these markers, must be open chromatin, otherwise it's closed. So those information are very useful because you can know for any given cells or population of cells, you know which region is open and what genes are in there. That information will tell you the states or function of the cells. So to detect those opening and what type of opening, there's a strategy, and there can be root back to people use so-called chromatin immunoprecipitation, ChIP-seq. In this study, you have this chromatin structure, this gray curve, and on this curve, chromatin, and you have different protein, okay, transparent factors, or by itself you have quite a lot of the histone to stabilize the structure of chromatin. And those proteins, if they are transcribing factors, they bind to the particular region and to regulate genes neighbor. If you, for example, are interested in this particular transcriptional factor with this triangle on top, what you can do is first you use physical approach, sonication, for instance, to break up all these chromatin structures. Especially those euchromatin regions will be broken, and make them into a small fragments of those DNA. And on each DNA, there still have this binding protein on there. Then you design a specific antibody. This antibody will be specifically designed to attach to this particular given transcription factor protein. this combination will help you fish out all the fragment which has been attached to this particular transcription factor. So after the binding, you can separate those particular region of the chromatin which connected or associated with this particular transcription factor you are interested in. So once you separate this with the rest, you can start to do the sequencing. The sequencing will align onto the region which this transcription factor will bind to. So you map back those regions onto the genome. So this region you mapped quite a lot. That indicates in your system, at that given moment, this particular transcription factor has a binding to that particular region in the genome. So this is the procedure. You do this binding and antibody binding, then use immunoprecipitation to find out, to enrich those binding fragments, then sequence them, and those DNA will be remapped to the genome. Then you know which part of the genome this particular protein transcription factor has been bound to. Okay? So here are the fragments you sequence, and then you look at the coverage. And in this place, you can do a smoothing, and you think, okay, here we have a peak. This peak is located in front of this another gene, so you know the transcription factor has been bound to here, and there may be a relationship in order to regulate the downstream, the gene next to this peak. So in ChIP-seq analysis, we try to identify the place where you show a peak and try to locate the binding sites of the transcription factor on the genome. So those are the picture of some of this ChIP-seq binding, and you probably use different transcription factors. So blue transcription factor, you do this for different samples, and these are different. So those information can be combined and to work out the relationship between the transcription factor and the binding sites and the downstream targets. So all built together, and also you can compare between different samples to see where you have a peak, where you don't have that, although they fall from the same transcription factor but different conditions of cells. So those studies. Okay, so this idea is for any given transcription factors, but you have to design antibody for many different transcription factors in order to achieve a global picture of multiple transcription factors activity, because one ChIP-seq only get the information for one transcription factor. In human and the mouse, for example, there are thousands of the transcription factors. There's no way you can do them all. So another technique is called open chromatin analysis. In this case, you don't consider any bindings, but you just look at the chromatin structure and try to estimate which part is opening, which part is closed. What you can do is to use the DNAs. So those are the enzymes we will cut open chromatin. So if you have open region, this enzyme will cut those into short fragments. Well, this closed part will not be cut. And after apply this partial digestion, sometimes you get quite a lot of fragments. Once you sequence them, then you know those blue fragments are located in this region of the genome. So you know this region must be open, okay, because you cut and get a lot of fragments that can be sequenced. Well, ATAC-seq is one of this group of the technology, but it's built into another enzyme which produced by the bacteria. This bacteria is very powerful to produce this ATAC-seq enzyme, and they can cut very sensitively to the small regions. So you get a high resolution of this position of the open chromatin using ATAC-seq compared to DNAs or itself. So gradually, ATAC-seq become a mainstream of the open chromatin structure analysis. So you can apply this and get genome-wide information, not only for any given information for any given transcription factors, but it's just global structure. This is open, and this is closed, this is open, to get that information very efficiently. So the enzyme is the transposes access chromatin structure. So this is a name. Those are the transposes. And those are the closed, open, and you get a fragment information from those open regions. Okay, you use next generation sequence to sequence a lot to get the FASTQ files and map back to the genome, so you know where is open chromatin, where is not. So 10x Genomics developed this technology to allow you to do this at a single cell level. So you extract nuclears from the individual cells and give to this machine, and inside the reagents you have those steps and to do open chromatin structure this IC for each cell. Okay, so when we see single cell ATAC-seq, is in reality they're single nuclears. Okay, and then you have those information for the position of the peak. So this is an example of single cell ATAC-seq data. So here, each row will be a cell, and then once you have the peak in that cell, you show non-zero values. Okay, this is cross chromosomes. This is probably just one small region, and you have so many different positions. So this huge matrix will show this cell in this region, for example, chromosome 3, 1568 position to 1575 position. So this region, there's how many reads in there. So you can get this very sparse matrix, contains a lot of zeros, but when you combine them, multiple cells, you will see the peak. Okay, this peak people found is consistent with bulk RNA-seq if you put multiple cells together. So this region is relatively small, but you can see there's so many different genes already under this region, and some of those peaks are in front, some of different genes. They are potential transcription binding site, potential regulatory regions, and at least we know this position is open. This position is open. This is in front of those genes. So those information will be very useful for you to get the global information about the cell states in terms of these open chromatin structures. So when you combine this information with real gene expression data at the same time, if you apply single-cell RNA-seq, ATAC-seq at the same time to the same population of cells, then you can start to use manifold alignment, because you get two matrices, one from expression of the genes, another are the open chromatin yes or no answer for each cell for each position. Although these two matrices are in a different size, but you just assume because they from the same population of cells, the cell are dispersed into shared common manifold. So if you align those cells into the same manifold, then those signals should be matched. But we don't know how to do it, actually. So this is the product page. I just get a screenshot. You can do single-cell ATAC-seq for your sample. You can also nowadays to do single cell RNA-seq and the single cell ATAC-seq in the same sample, same cell. Okay. This is more powerful because you don't even have to align, because you get the same information about all the genes expression for these cells. At the same time, you get the information of all the open chromatin information from this particular cell as well. I think we still have time to play a small video here.
Transformative advances in science require multidimensional approaches to more deeply explore underlying biology. Enhance your characterization of cell types and states to uncover previously hidden insights. Access a unified view of transcription and the chromatin landscape by combining gene expression and ATAC-seq data from the same single cell. Open chromatin regions are key to regulating gene expression. Identify cells with similar gene expression profiles and layer on chromatin landscapes. Better resolve cell states using this combination of modalities. Link gene expression to accessible chromatin regions to understand how genes are regulated and cell types and identities are established. Leverage two modalities in one simple workflow, using the first commercial multi-omic product to simultaneously capture the transcriptome and epigenome from the same cell. Transform your view of cells, tissues, and beyond. Multiply your power of discovery with multi-omics.
Okay, so this is something you can consider if you help your PI to design any single cell related projects. Let them to consider and to go beyond the expression, but also have this multi-omics. So we can even go more those days if you have the protocols to do, for example, not only chromatin structure, but also methylation is possible if you know how to do it. You can sequence the DNA, it's possible, okay? The genome. If you believe this CNVs are maybe important in the cancer cells, for example. You can also label some surface protein, and their established commercialized 10x genomics kits allow you to see, I want to do single cell RNA-seq expression for that cell. At the same time, I want to see the surface protein expression level. And the T cell receptors, so it's kind of similar multi-omics we mentioned last time. You have a T cell expression and their T cell receptor profiling the types, or P cell, B cells. So they all belong to this multi-omics integration. But this field, if you gave me the data, I don't know how to analyze them to extract the most valuable information. Still trying to learn this. We don't have established solutions. There's still a lot of things under development. I think recently, Surite have this extension to see, okay, we can use Surite to process single-cell ATAC-seq data, okay? Basically, it's doing the same thing as a single-cell RNA-seq, but the elements, the values in this matrix, single-cell ATAC-seq matrix, is not the expression level, just the count of open chromatin the counts for that region, for that cells. And it's more sparse and contains more peaks numbers than number of genes. Okay. But they believe they can, for example, use single-cell ATAC-seq to do the dimensionality reduction, can do t-SNE, UMAP, and you can achieve almost the same results as you have a single-cell RNA-seq expression. So here you don't use expression information. You use whether the cell has this peak, or that peak. Another cell has this peak, that peak, this peak. The combination of those peaks give you the same information about the expression. So you still can do the cell type identification to do DE analysis, but it's based on open chromatin counts. And the video I just played, it have a multi-omics. You have a single cell RNA-seq, you get the population of the cluster of one cell type, but you apply ATAC-seq information, you can separate one cluster of cell into two separate subclusters because they have a same expression matrix, but the ATAC-seq gave you more detailed resolution to find the substructures within subclusters. I think from Surit version 3, they start to have those capacity to deal with single-cell ATAC-seq. Our toolbox, my toolbox, I don't have this ability to play with both. Okay, so we jump to another topic. Before that, I need quick questions from you. Mm-hmm. Mm-hmm. I don't think the current technology, or I know the protocols can handle that. It's very complicated. You have mitochondria, you have viral infections. It's not possible at this moment. Yeah. Okay, tensor of rank 3. So you have a matrix of one layer, and then you have a matrix of another layer, you have another layer. So this is a high order data structure. You can consider this as a high dimensional matrix, 3D array. So you basically you have this, and you have a more realistic picture. So this introduces 3D. The current algorithm or the computer framework are really not very good to handle anything which is beyond 2D. Or matrix, we can play so many algorithms, operations. But once you reach to 3D, I don't know even how to easily to represent or handle it in the memory. So not even 1 to 3D, you can go even higher, okay? Consider tensor structure. It's higher dimensional. Well, so what type of data is high dimensional data? The image itself is, so the color image, so like this there. If you look at how the matrix data is stored, it actually have 3 matrices, each for 3 different colors and the density of 3 different colors. So when these 3 are combined, you have this picture. You can convert this into the gray scales. In that case, you only have a black and white. In that case, you have a matrix, but in general, those color pictures, it's 3D matrix. So image itself is like this. For this particular training data, for example, and you have handwriting numbers, and each picture is 180 by 180 no color matrix, but if you put the training data as a vector, so you have this handwriting pictures, then you give this 3D data, a vector, 3D tensor to the training. So this is like a third order tensor. There are many, many others, okay? Video. You have a color, you have a frame at a different time points, okay? So this is a 4D. So there are many, many. This will be recommender system, the Netflix viewer and the movie, but this is Monday, Tuesday, Thursday, they're all different. So you have this high dimensional structure. So we have quite a lot of examples in the real applications. Physics, okay, they use tensor in their equations all the time. All these symbols are tensors. They have space, location, and also time. So they have to put that tensor operations in those equations. As you can imagine, if you design an algorithm to handle the tensor, you have to do some kind of loop. You have x, y, and z. Okay, so this increases the complexity of the coding. In this particular example, you have a tensor that's 2-3 layers, 1, 0, 1, 2, and yeah. So for different colors, so you have to be able to handle this. And in computer science, people call number array, 2D array, ND array. In mathematics, you have scalar, vector, matrix, or tensor. So same terminology, but you can use them interchangeably, but based on the field. So as we mentioned, that eigen decomposition, matrix decomposition, we can do the tensor decomposition. Okay, this is one of the powerful way to learn the different components from a tensor. Okay, huge tensor structure. You learn what's the major components from the data itself, okay? So you want to do the decomposition to learn those major contributors. So if you have a 2D matrix, then you can decompose it into two vectors. This outer product will create this matrix. If not accurate, you can add another 2D. So this is decomposition. Same idea for the three-way tensor. In this case, you have to have outer products from 3 vectors. So A and B multiplication and make outer product become a matrix. This matrix will be multiplied with another vector C and create this 3D matrix. You can add this up to do this decomposition for even higher dimensionality data sets. On your left, this is SVD, if you refer back to our 2 weeks early. So you can decompose a matrix into U sigma V, and you can use those top singular value composition and to reconstruct or approximate original. So this matrix, which skinny compared to the original, it's only 2 major dimension, will multiplied by this matrix contains singular values and multiplied by this V transpose. This will approximate the original. So same thing, you can decompose a tensor, a high-dimensional structure, into 1, 2, 3, 4, this component, and plus some kind of noise. And what you learned from those matrix here are the major components which you can do the multiplication to reconstruct the original, the whole set of data. This is so this SVD structure. Similarly, this will be the matrix decomposition. So this NMF matrix factorization. So you can have these two vectors and multiplication, and then you put them together, you will approximate the original. Very similar, you can do the tensor factorization, not a matrix, but here you just directly using algorithm to estimate those major components, and the multiplication of them reflect to, if you put them together, these major components will reconstruct a similar value compared to the observed. What we are interested in are the values in those vectors, because they reflect what are the major components of these vectors, contribute to explain those. Some of those vectors, the values, the change, will teach you some of these underlying principles or mechanism to create the observed tensor structure data. Yeah, so we are interested in the values in those components. So this method have the name so called CP. So if you see CP, people know it's a canonical decomposition. They also sometimes call it differently, but they're very complex. The history of those development in the numerical approximation field. So the problem we want to get is essentially it's an optimization problem. We want to estimate the values for those vectors. Okay, we don't use many. We just use a small number of those vectors, and we want to estimate the values for each of those components. And our goal is to, after this operation, you get the as close as possible approximation of the original or observed data in its tensor form. So the objective function is try to reconstruct the tensor to make the difference between observed and the reconstructed tensor, the value, as smaller as possible. This is like a norm between them. So the M is achieved by different components, vectors, multiplication together. So this part is M. This is X itself. The current algorithm people use most, I believe, this so-called fitting CP is called alternating least squares. It will fix one dimension first. If you have a 3D, it will start to iterate I, and then move to another step to iterate J. You have ijk 3 dimension. and then you alternate to another dimensionality and a k. So you do this 1 dimension, 2 dimension, 3 alternating. Try to estimate, update the values within these vectors in order to achieve at the end the approximation. So you do this by a loop until you see the difference between them are small enough, I stop. So people try to reduce the number of this looping, because if you have, say, millions of loops in order to achieve some of this, to take too long, is you have to have a strategy to reduce the number of looping to make this converged very fast. There's some strategy. I don't know exactly, but that's the goal. Try to use some shortcut to minimize the computational loading and the computational task. One strategy, I believe they are using this some subsampling instead of using the all, but subsample some of this, some tricks. So there's another different ways to do this decomposition. So CP is one of the a school of the way of direction. Another way is called Tucker. Okay, Tucker is to decompose into the 3D tensor decomposes into 3 mode of these fibers, and then the product of them reconstruct the original data. So applications, one of this is in this YouTube, there are a good illustration about those cell phone use in a school and who's calling who at a different time. So you can imagine if you sample every 2 hours across the day or across the whole week, then you have a network of the communication, and then for each time point, but because you have multiple time points, and this create a very sparse binary who's calling who at which time. So you have this network information, but it's a multi-layered network. And then they apply this algorithm and to see what's the components, and they learn the receiver components, sender components, and the time and the day. I believe they have a relatively long term across the whole semester, maybe. So those information, once you combine them, you can somehow reconstruct the whole network across time. And those values in those different components, they use how many? 12 components. Okay, some components discover the peak use, maybe communication in different days, and what's the peak. So somehow they give you some insights about this network involvement. Okay. This is another interesting setup they have. I think in this case they put the mouse in the center here and let it to go left and right, and then their food reward if they go the right direction. then at the same time, they record the neuron activities for the mouse when they move to the maze. And also this is a time framework, and also multiple trials and it replicates. So at the end, you have this very complex neuron by time and by trial, this tensor information. And then they learned the neuron activity components. They learned the time and also the trials. What they can find is some of the signals are associated with reward, some of the associated with left turn, right turn. So once you decompose those, then they are not just a collection of data. They start to show the major patterns, and the sum of the patterns may have biological meaning. Some of the neurons, they can say, okay, this neuron associated with left turn, and this neuron associated with right turn, and this neuron associated with reward once given. So they start to discover those differences. Yeah, so if you are interested in this video, starting time is here, so it's very insightful. Yeah, so the last slide is our own applications. I don't remember, I have explained this to you or not. This is the package we developed in order to compare two samples, and for each sample we create gene regulatory networks. They used, so this is one sample, another. Compare these two rather than using differential expression. We create networks first, and we subsampled, basically subsampled the cells, randomly sample, say, 1,000 cells out of many of these, and they construct replicates of random subsampled data. And for each subsampled data, we create a gene regulatory network. So it depends on how many genes you have. In this case, you have n by n genes. Okay, if you can see this n by n genes. And because we subsampled t times, so you have n by n by t. So this will be a tensor structure. For another sample, we also have this 3D structure, network between genes and the subsampled. Okay, so this is 3D. We put them together, will become a 4D. We gave this to the tensor decomposition algorithm. Then to learn these major components, then we pick top components to reconstruct the whole thing, reconstruct the tensor. In that case, some of the noise will be removed because we smooth out, only learn the major components. And then take average across this smooth or denoised version and get two gene regulatory networks. So each from one sample, black and red. Then we consider these two networks as each gene, the properties is defined by its interaction with other genes. Basically, this look at as a profile of each genes. Then we believe these two gene sets, they come from the same common manifold. So we did this manifold alignment to try to project the gene from a black and the red samples onto same manifold. If they are highly similar, then all the genes should be projected onto same very similar location, one from black, one from the red. If these two networks are very different, then after the projection, some of the genes will far away from each other, okay? Because we have the same gene from black, same gene from red. So these pairs of the red, black of the same gene, most of them should be aligned in the common manifold nicely, so we don't see the difference. But we want to detect those genes which after alignment they cannot be put together. So those genes will define the difference between these two networks. It's defined by their interaction with other genes are different from each other. So we found those genes and prioritize a short list for that genes for downstream analysis. See, what are those genes that cannot be aligned considering the manifold? So those genes we found is very useful for most of the genes are not differentially expressed. They have a similar average expression between these two samples, but their interaction pattern with other genes changed greatly. So we call this differentially regulated genes and make a package. So it's allow people to compare two samples, but not just directly compare the mean, but to look at the differentially regulated genes between two samples. So those are application of the tensor decomposition for denoise purpose, and also there's a manifold alignment in order to learn the nonlinear relationship between genes in the two networks. Okay, so that's all for today. We still have time if you have questions. Okay, I also posted this new homework. If you have any questions, let me know. This will be your last homework for the semester. I also have this your presentation timetable. Some of these you will present on Tuesday, some will be Thursday. We will be in different rooms. Tuesday will be here, Thursday will be there. So look at your the data of your presentation to think about whether you will be standing here or there. They're slightly different setup. Okay. All right. Thank you.